Have you ever like really thought about, I don't know, the hidden like inner workings
Mm.
behind a flower?
Yeah.
I mean, or how life just keeps, you know, get generation after generation. Like, how does that actually happen? It seems so simple on the surface, but once you start digging in, especially with plants,
Right. It's mind-boggling.
it's way more complex than it looks.
Definitely. That's what we are going to unpack today
Yeah.
in this deep dive
Yeah, in this deep dive.
into the fascinating world of sexual reproduction in flowering plants.
Right. And we're going to be working off of a very detailed biological study for this one.
Mm-hmm.
And so our mission here, I think, is to like extract the most important, most interesting aspects of this whole process for you, right?
Right.
To give you a shortcut to understanding like the fundamental biology of plant life,
Exactly.
And you shared this material because you wanted a clearer understanding of this, like how it actually works.
Yeah.
And we're here to, guide you through those crucial elements.
Yeah.
But like taking a step back for a second, I mean, it is really amazing how life, you know, at the species level persists
It is.
through reproduction,
Yeah.
right? Like you and I, in individual organisms, we have a limited lifespan,
Yeah.
right? But our genetic information, it gets passed on,
Right. Yeah.
And sexual reproduction is really a cornerstone of that,
Right.
because you get this mixing of genetic material, which creates variation within a species.
Yeah. It's key.
And that's what allows the species to really adapt and survive in an ever-changing environment.
For sure.
And this happens in, flowering plants, it happens in humans. It's
Yeah.
like a fundamental biological process.
A common thread through all living things, pretty much.
Now, our source mentioned this person, Panchanan Maheshwari.
Oh, yeah.
I've never heard of him before.
Yeah.
But he's an incredibly influential botanist from India.
That's right. A real pioneer.
His work in plant embryology, taxonomy was just truly groundbreaking.
Very foundational work. Yeah.
He helped develop some of the early biology textbooks
Wow.
in India. So I always like to think about like the human stories behind the science.
Absolutely. Yeah.
It's really interesting. It adds so much,
Yeah.
to just the cold facts.
Yeah. And Maheshwari, I mean, his contributions really underscore the depth and richness of botanical science.
Absolutely.
So, all right. So we appreciate the flowers. They're beautiful.
Right.
The fragrance, the colors.
They're amazing.
It's easy to just think about that,
Right.
But our source really drives home the point that all those qualities, they're not just for our benefit.
No.
They're all adaptations specifically for sexual reproduction.
It's true.
Which is crucial to the plant's survival.
That's how they perpetuate themselves.
Yeah, it's not just to please us, aesthetically.
No, they're not putting on a show for humans exactly. Right. All right, so let's get down to it. To a biologist, a flower is a morphological and embryological marvel, our source says.
It really is.
Right? It's a site for sexual reproduction. And I know we all vaguely remember the parts of a flower from like grade school, petals and stems and all that.
Yeah.
But the real action, like where the magic happens, is in a couple of key structures, right?
Yeah, it's not the parts you necessarily think about when you first see a flower.
Right.
So we've got the androecium.
Okay.
That's all the male parts.
Okay.
And the gynoecium.
Okay.
The female parts. And these are where those specialized structures and cells needed for reproduction actually develop.
Okay.
So these are the key players.
Okay. Now, before fertilization can even happen, there's this whole sequence of events, right? This pre-fertilization. It's not just like, boom, instant.
Well, no, it's a whole process.
Right. What's going on there?
So the plant has to decide to flower.
Right.
And that involves all sorts of internal signals, hormones shifting structural changes within the plant.
Okay.
And this leads to the formation of the floral primordium, which is like, the baby bud.
Okay.
That's going to become a flower or a cluster of flowers.
Okay, got it.
Which we call an inflorescence.
Okay.
And within those developing flowers is where those male and female reproductive structures start to form.
Got it.
So the androecium containing the stamens.
Okay.
And the gynoecium containing the pistils.
Okay.
So those are the male and female parts.
So let's focus on the male side first then.
All right.
The androecium is basically a bunch of stamens.
Mm-hmm.
What does a stamen look like?
Okay, so a typical stamen has two main parts.
Okay.
You've got the filament.
Okay.
That's like the stalk.
Okay.
And the anther at the top, which produces the pollen.
Okay.
The filament's like the support attaching the anther to the base of the flower. Now, what's interesting, and this is where it gets kind of cool, is that the number and length of those stamens can vary like crazy
Mm.
between different plant species,
Oh. Oh, wow.
which reflects their different pollination strategies.
Oh.
Like they've evolved in different ways.
Okay, fascinating. All right. And inside the anther, this is where the pollen's
This
produced.
is where the magic happens, yeah.
Yeah. Our source mentions that a typical angiosperm anther is bilobed and dithecous. And I have to admit, I don't remember what those words mean.
Right.
Can you break that down?
Okay, so bilobed means it has two lobes, two distinct sections.
Okay.
And dithecous means each of those lobes has two the theca, which are basically compartments or sacs.
Okay.
So if you cut across an anther, you'd see this four-sided structure with four pollen sacs inside,
Okay.
two in each lobe.
Got it.
So we call them microsporangium.
Microsporangia.
And it's a pretty common setup in flowering plants.
Interesting. So each of these pollen sacs, they have their own structure too.
Yes, they're kind of like little fortresses.
Okay.
So each microsporangium,
Okay.
it's surrounded by four wall layers,
Oh,
like protective barriers.
okay.
The outermost layer is the epidermis,
Okay.
which, offers that first layer of protection.
Right.
And then right beneath that is the endothecium.
Okay.
And that plays a huge role in dehiscence,
Okay.
which is the fancy term for when the anther splits open
Okay.
to release the pollen.
Okay.
And then there are one or more middle layers,
Okay.
which don't last very long,
Okay.
but they help nourish the developing pollen.
Okay.
And then finally,
Mm-hmm.
the innermost layer, the most important,
Okay.
the tapetum.
Okay.
The tapetum's super important because it provides nourishment to the developing pollen grains.
Oh, wow. Okay.
It's like their personal chef.
Okay.
These tapetum cells are unique.
Okay.
They've got this dense cytoplasm, often multiple nuclei. They're like little powerhouses.
Okay.
And at the very heart of each young microsporangium, you'll find these densely packed cells called the sporogenous tissue.
Okay.
So that's going to be the pollen eventually.
Okay, so we got the sporogenous tissue. What's the next step?
All right, this is where microsporogenesis comes in.
Okay.
So as the anther matures, the cells of that sporogenous tissue, which are diploid, meaning they have two sets of chromosomes,
Okay.
they undergo meiosis.
Okay.
Now, meiosis is a special type of cell division that cuts the chromosome number in half.
Okay.
So each of these cells, also called a pollen mother cell,
Okay.
divides to form a cluster of 4 haploid microspores.
Okay.
So now they've got only one set of chromosomes
Okay.
and this little group of 4.
Yeah.
We call it a microspore tetrad.
Okay.
And at first they're all stuck together,
Right.
but as the anthers dry out,
Yeah.
these microspores separate.
Okay.
And each one develops into a mature pollen grain. And it's incredible the sheer number of these pollen grains.
Wow.
Thousands
Wow.
within each microsporangium.
Okay.
It's all about maximizing the chance of successful pollination.
Oh, okay, that makes sense. All right, so now we have these fully developed pollen grains.
Right.
And this represents the male gamma-phyte.
Right.
The structure that produces the male gametes.
Mm-hmm.
So what does a mature pollen grain look like?
It's basically a tiny sphere,
Okay.
like 25 to 50 micrometers in diameter, super small.
Okay.
It's got this distinctive 2-layered wall.
Okay.
The outer layer, we call it the exine.
Okay.
That's the tough guy.
Okay.
It's made of sporopollenin.
Okay.
It's like nature's Teflon.
Okay.
It can withstand crazy temperatures,
Wow.
acids, even enzymes that break down other things.
Wow.
Which is why we can find pollen grains in fossils like millions of years old.
That's amazing.
That's pretty cool.
And it's not just a plain shell, is it?
No, it's got these openings called germ pores. It's where the sporopollenin's missing.
Okay.
And these pores are important later on for pollen germination.
Oh,
And get this, the exine has all these intricate patterns
Mm.
that are often unique to different plant species.
So you can tell what plant it came from just by looking at the pattern?
Often, yes. Like a fingerprint, almost.
Wow.
We can use it for identification.
That's amazing. All right. So that tough outer layer, the exine,
Yeah,
it's all about protecting
absolutely.
those delicate contents inside.
Protecting that precious cargo.
Yeah. And those germ pores have this specific purpose
Yeah.
that we'll get to.
They'll come in handy later.
Yeah. Okay.
Yeah.
What about the inner layer then?
Okay, so the inner layer is called the intime.
In time.
It's much thinner,
Okay.
and it's made of cellulose and pectin, like plant cell walls.
Okay.
And inside the intine is the pollen grains cytoplasm
Okay.
with its plasma membrane.
Okay.
Now, when that pollen grain matures,
Mm-hmm.
it usually contains two cells,
Mm-hmm.
a big vegetative cell
Okay.
and a smaller generative cell
Okay.
nestled inside.
Okay. Got it. And these two cells, I'm assuming they have different roles to play.
Yeah, they're a team,
Okay.
but each one has its own specialty.
Okay.
So the vegetative cell, it's the big one.
Right.
It's got this large nucleus
Okay.
and lots of stored food.
Okay.
It's going to form the pollen tube.
Oh, okay.
That's the one that carries the male gametes to the ovule
Okay.
and the generative cell.
Right.
That's the smaller one.
Okay.
It's spindle-shaped. It's got this dense cytoplasm in its own nucleus.
Okay.
Now, in over 60% of flowering plants, the pollen grains actually released at this two-celled stage.
Okay.
But in some species,
Mm-hmm.
that generative cell divides
Okay.
before it's released
Okay.
to produce two male gametes.
Okay.
So those are released at a three-celled stage.
Okay.
So we've got these 2 or 3 cells,
Yeah.
and they represent the male gametophyte. You know, it's the structure that either produces
Yeah.
or already contains the male gametes.
Okay. Got it.
Yeah.
It's amazing how much is going on at this microscopic level.
It is.
You know, it's like a whole little
a
world.
whole universe inside that little grain.
But, you know, pollen isn't always a welcome guest.
Right.
I mean,
Yeah.
a lot of people suffer from allergies,
Oh, for sure. Yeah.
pollen allergies,
Big time.
and it can really, you know, cause a lot of problems.
Absolutely. And it's funny because you think of pollen as this beautiful thing.
Right.
Right, because it's creating life.
Yeah.
But for some people, it's a real nuisance.
Right.
And it's not just any pollen, it's certain types
Right.
that trigger allergic reaction.
Right.
And these reactions, they can range from just, mild sneezing,
Yeah.
itchy eyes,
Yeah.
to some pretty serious respiratory problems like asthma or bronchitis.
Right. And once all these pollen grains are released,
Mm-hmm.
you know, they have this limited time to reach the female part of the flower.
Right, they're on a mission.
Before they lose their mojo.
Exactly.
So how long can they typically stay viable, you know, able to fertilize?
Well, that really depends on the species
Okay.
and the environment.
Okay.
Like temperature, humidity,
Right.
it varies a lot.
Yeah.
I mean, in some plants, like rice or wheat, those pollen grains might only last like 30 minutes after they're released.
Wow.
But then on the flip side, you've got some species in the rose family or legumes
Mm-hmm.
where the pollen can stay viable for weeks,
Wow.
even months.
Wow. It's pretty amazing. And you know how we can freeze sperm, like preserve it?
Oh, yeah,
Can we do that with pollen?
we can.
Wow.
Yeah, we can freeze those pollen grains
Really?
from a lot of species and store them for a long time,
Oh, wow.
years even.
Wow.
And it's done by freezing them in liquid nitrogen
Okay.
at like super low temperatures. And then they're stored in these pollen banks.
Oh, wow.
Like seed banks.
Oh, wow. That's so cool.
Yeah, it's a really valuable resource for like crop breeding programs. It's
Ah.
amazing what we can do now.
That's really cool. All right. So we've covered the male reproductive side in detail.
Right.
You know, let's move on to the female reproductive part of the flower, the gynoecium.
Okay, let's head over to the lady parts.
So the gynoecium, this is made up of one or more pistils, right? And what's a pistil again?
Okay, so if it's just one pistil, it's called monocarpellary.
Okay.
And if there's multiple,
Mm-hmm.
it's multicarpellary.
Okay.
Now, those multiple pistils,
Yeah.
they can be fused together,
Okay.
which is called syncarpus,
Okay.
or separate,
Okay.
free from each other,
Okay.
which is called apocarpus.
Okay.
A little bit of terminology there.
Right. Got it. Got it.
But basically,
Yeah.
each pistil,
Okay.
whether it's by itself or in a group, it typically has 3 parts.
Okay.
You've got the stigma at the top,
Mm-hmm.
which you know is that sticky surface that receives the pollen.
Right. Right.
You've got the style in the middle,
Okay.
like a stalk,
Okay.
and the ovary at the base.
Okay.
And the ovary contains
Yeah.
the ovules,
Okay.
also known as megasporangia.
Okay.
So that's where the eggs develop.
Okay. And the number of those ovules.
Yeah.
That can vary a lot too.
It really does.
Right.
Yeah.
I mean, some plants like wheat, rice, mango, they might only have one ovule per ovary.
Right.
Right. But others,
Yeah.
like papaya, watermelon,
Mm-hmm.
orchids,
Yeah.
they've got tons of ovules.
Lots and lots.
So, and that directly affects like how many seeds
Right.
are in the fruit. Right.
Exactly. More ovules, more seeds.
Okay. Got it. Got it.
Yeah.
All right. So the ovule or megasporangium,
Yeah.
It's pretty complex, too.
Yeah.
It's not just a simple little...
No, it's a world of its own. It's a small structure,
Okay.
but it's got all these different parts.
Okay.
So first,
Okay.
it's attached to the placenta, the part of the ovary,
Okay.
by a stalk called the funicle,
Okay.
and the point where they're attached.
Yeah.
That's called the hilum.
Okay.
And then each ovule is typically surrounded by one or two protective layers
Okay.
called integuments.
Okay.
And they enclose the central part of the ovule, but they don't cover it completely.
Okay.
So there's a little opening at one end called the micropyle.
Okay.
And opposite that
Yeah.
is the chalaza, which is basically the base.
Okay.
Now, inside those integuments, we have the nucellus, which is like a mass of tissue that's got stored food.
Okay.
And then inside that is the embryo sac.
Okay.
That's the female gametophyte.
Okay.
So that's where the egg cell is.
Okay.
And usually only one embryo sac develops
Okay.
from a megaspore within each ovule.
Okay, got it. So just like we had microspore genesis to make pollen,
Yeah.
there's a corresponding process to form these megaspores, right?
Exactly.
Okay. So what's that called?
It's called megasporogenesis.
Okay.
And usually what happens is a single large diploid cell, the megaspore mother cell, we call it the MMC,
Okay.
it differentiates in the ovule.
Okay.
It's got lots of cytoplasm and a prominent nucleus.
Okay.
And then it goes through meiosis,
Okay.
that reduction division,
Right.
to produce 4 haploid megaspores,
Okay.
which are arranged in a line
Okay.
called a tetrad.
Okay.
So just like with the microspores, they start out as a group of 4.
Got it. Got it. Okay. And then one of those becomes the embryo sac, right?
Right, yeah.
So how does that happen?
Usually only one of those 4 megaspores survives.
Okay.
The others, they kind of just wither away.
Okay.
And that surviving megaspore, it's the chosen one.
Okay.
It develops into the female gametophyte, the embryo sac. And this particular type of development, where it comes from a single megaspore, it's called monosporic development.
Okay.
And it's the most common way it happens in flowering plants.
Interesting. Okay. So we've got this one functional megaspore, right? And it goes through this amazing transformation
Yeah,
to become this mature embryo sac.
right.
Our source mentioned it's 7-celled but 8-nucleate.
Right, which sounds kind of weird.
Yeah.
Right.
Yeah. I mean, how does that work?
Okay, so the nucleus of that megaspore,
Yeah.
it goes through 3 rounds of division,
Okay.
mitosis this time.
Okay.
So the first division makes 2 nuclei
Okay.
and move to opposite ends.
Okay.
So we have a 2-nucleate embryo sac.
Okay.
Then those nuclei divide again.
Mhm.
That's 4.
Okay.
And then one more time.
Okay.
8 nuclei.
Okay.
And here's the interesting part.
Mm-hmm.
At first, those divisions are what we call free nuclear.
Okay.
So the nucleus divides, but there aren't cell walls forming right away.
Oh, okay.
It's just this big sac with 8 nuclei.
Okay. Wow.
Then, once we reach that 8-nucleate stage,
Yeah.
finally the cell walls form,
Okay.
and we end up with this highly organized, mature embryo sac.
Okay. So what does that final arrangement look like?
Okay, so at the micropeeler end, remember that little opening we talked about?
Yeah.
There's a cluster of 3 cells called the egg apparatus,
Okay.
and this is where the egg cell hangs out.
Okay.
That's the female gamete, ready and waiting.
Okay.
Now, flanking the egg cell are 2 other cells called synergids.
Okay.
And they have these special thickenings at their tips called the filiform apparatus.
Okay.
It's almost like they're beckoning the pollen tube, like, come hither.
Right.
And at the opposite end, the chilazal end,
Mhm.
there are 3 more cells called antipodals.
Okay.
We don't really know what they do.
Right.
They often just degenerate.
Okay.
And then in the very center
Mm-hmm.
is a large central cell.
Okay.
And this one's special because it has 2 nuclei, the polar nuclei.
Okay.
So if you're counting, you have 3 cells at one end,
Yeah.
3 at the other,
Okay.
and 1 big one in the middle.
Okay.
That's 7 cells.
Okay.
But that central cell has 2 nuclei.
Right.
So that brings the total to 8.
Okay. I got it. So that's why it's 7-celled,
Yeah.
8-nucleate.
Why? It's unique.
Okay. Wow. So it's this super precise
Absolutely.
cellular architecture.
This has to be to work, right?
Yeah. I mean, okay. So now we have the male gametophyte,
Right.
the pollen grain,
Yep.
and the female gametophyte.
That embryo sac.
Right.
Yeah.
The embryo sac.
Yeah.
So we need to get them together, right?
Yes.
For fertilization.
Somehow.
That's where pollination comes in.
Exactly. Pollination's the bridge.
Okay.
Because these gametes,
Yeah.
they can't move on their own.
Right.
So we need to get the pollen from the anther to the stigma. That's what pollination is.
Okay.
It's the transfer.
Okay. And the way that happens, that can vary.
Oh, yeah.
right? I mean, you've got things like
Wind,
wind, yeah,
water,
water,
animals.
right, right. All different players.
Yeah.
So what are the different types of pollination then?
So based on where that pollen's coming from,
Yeah.
we can categorize pollination into three main types.
Okay.
First, we've got autogamy.
Autogamy.